Chapter 14, Section 8, Iron Oxidation. Now what we're going to be looking at here is an aerobic oxidation of ferrous iron, Fe2, to ferric, ferrous to ferric iron, Fe3. Okay, this is going to be done by chemolithotrophic iron bacteria. Now, they're not taking up the iron into the cell so much as they are stripping off an electron. Notice 2 plus, 3 plus. That means it is here it's heavy 2 protons, here it is heavy 3 protons. Light 2 electrons, light 3 electrons. In the process of doing this, there's going to be the production of ferric hydroxide, Fe3OH, and I can't remember how many OHs, but we'll see in a slide or two. In the process of doing this, there's going to be free hydrogens released, which means pH is going to be dropping. So this is going to be for bacteria that are highly acidophilic. And you see here, this is showing that in this water here, that it's looking, this isn't mud, this is actually ferric oxide rust. So this water looks rusty because FeOH3, there's the 3 hydroxides to the iron, and then it's highly acidic. Here's your rust, here's your acid. Excuse me. Examples of these iron bacteria: Acidothiobacillus and Leptospirillum. These grow autotrophically, meaning they do their own manufacture. They do their own carbon fixation. And they can grow as low as pH 1, but normally pH 2 to 3. Basically, stomach acid is where they grow the best. They've isolated these in acid-polluted environments. Primarily a really good place if you ever want to go and isolate these. Go look in water's runoff from coal mines. A lot of my family is from West Virginia, up in Appalachia, who are coal miners going back generations. So go up and visit. And yeah, there's a lot of the water there stinks because it's got a lot of sulfur in it in some areas. In other areas, it looks rusty, muddy, until you get down and look at it, and it's actually not mud, soil, it's rust. And it stinks, and it stings when you get it on you. So it's bacteria similar to this growing in that mine runoff. Now, if you take these bacteria, put them in a near neutral pH water that's, quote unquote normal, you're going to get the iron, the ferric iron, ferrous iron, Fe2, transitioning anoxic to oxic, low level or no oxygen to oxygenated conditions. Well, during this time, the bacteria are going to be able to go oxidize iron before the water becomes oxic, and whatever iron is in the water will spontaneously oxidize on its own, irregardless or regardless of whatever bacteria may or may not be present. When you look at the respiratory chain, remember this is the cytochrome complex proteins that are going to be forming an electron transport chain that sets up a proton mode of force that drives ATP synthase. That's the respiratory chain here. So the bacteria are going to have their own cytochromes containing a protein known as rustacyanin. This is a periplasmic copper-containing protein, means it's going to be between the outer and the cytoplasmic membrane in that periplasm. So we're talking gram-negative bacteria. Copper-containing, so remember the cytochromes are going to have the different ring structures. Well, here it's going to have copper at the center as opposed to iron or magnesium, as we've seen in previous sections. The iron oxidation begins at the outer membrane. So the iron never actually gets into the cell. As we're going to see on the next slide, we get the electron stripped off, oxidation there, and then it is passed through a series of proteins from the outer membrane through the periplasm through the cytoplasmic membrane. One of those that's going to take it and pass it on is that rustacyanin that was mentioned before. So we go Fe2, ferrous to ferric, or ferric to ferrous. I always get those mixed up. Fe2 to Fe3, which is always we'll sit there and talk about them. Yes, I know all the chemistry majors and who are listening to this, their heads just blew up. How do you not know this? I don't study iron oxidation. I don't study iron metabolism that much. So Fe2 to Fe3, remember this is nothing more than stripping off an electron. That electron is going to be transported across the outer membrane using a cytochrome C protein. It's going to be passed on to the rustacyanin, which is in the periplasm. That electron can take one of two paths. First path, passed on to cytochrome BC1, that is going to take it and is going to utilize it in that reverse electron flow, which is going to take hydrogens, and instead of having them flow back through, it's going to pull them across, and in the process, it's going to reduce NAD to NADH. The other potential pathway it could take is rustacyanin and pass it on to cytochrome complexes that will do notice it's a very reduced one that will pump across hydrogens, setting up a proton motive force. Here, oxygen is the final electron acceptor, so these bacteria are aerophilic. The hydrogens will flow through ADP to ATP via this ATP synthase. Now, the NADH and the ATP that were just made, those are not for the normal, quote-unquote, normal cellular metabolism. No. What they're for is the Calvin cycle, taking gaseous carbon dioxide and fixing it via CO2 fixation, carbon dioxide fixation, to make short-chain sugars. Now, those short-chain sugars, the G3Ps can be joined together via gluconeogenesis or a version of to make sugars, carbohydrates, which can be utilized as energy or can be utilized for, peptidoglycan, whatever. Or G3P can be modified to become acetyl-CoA. The acetyl-CoAs can then be linked together 2 by 2 by 2 to make short-chain fatty acids, which will then be joined together as long-chain fatty acids, joined to a glycerol, which you now have your mono, di, and triglycerides, which any of those can then be formed into your phospholipids. You take the G3Ps, make them into glucose, G6P, take it through the shunt, and you now have your 5-carbon sugars. You can have nucleotides, and it just keeps going on. Same with amino acids and everything. The problem is this iron oxidation is a very poor energy yield. Okay, because think back a few sections ago where we were talking about, the E, the V, the E naught of these half reactions, where you have to couple them, this half of the reaction with another half. A plus B, yields C plus D. The A to the C, the B to the D. How those, have to be far apart on that one graph. What was it? Figure 14.1, I believe it was. That showed, as long as they're farther apart, Their EV values means that the energy yield from these reactions will be higher. Well, it turns out that the energy yield from iron fixation, yeah, not so much. So they have to go through and they have to oxidize large amounts of ferric iron to ferrous iron just to get the one electron at a time so they can power their own autotrophic metabolic needs. Now, it can be done under anoxic conditions. Remember, anoxic means lack of oxygen, or even if oxygen is there, not required, it's not going to be the final electron acceptor. Certain chemolithotrophs and phototrophs will do this. Fe2 is used as either the electron donor or some form of reductant for CO2 fixation. So it's either going to go Fe2 to Fe3, kicking off the electron like we've seen in the previous couple of slides, or during the process of CO2 fixation, there is going to be an iron giving up an electron and or accepting one as a means of allowing for CO2 to be joined together to form, a C2, C3, carbon chain. What we notice is that even though these bacteria that are using iron oxidation thrive at acidophilic pHs, near neutral pH, E0 values, as remember I just talked about that in the last slide, of this conversion, Fe3, Fe2, well, it's more electronegative, which means greater potential for yield due to the cytochrome C being in a better, stabler environment. And just a picture showing you growth and what is this? This is just the cultures, sterile, inoculated, and then this is showing you when the iron oxides are actually produced by the bacteria. So here's what the broth looked like before. Here's after the bacterial added, no growth as of yet. And then here's what it looks like when you come back at a later time point. This is where all that rusty water look comes from.